welcome back. So in my previous lecture, we have already discussed about the different types of the heterotrophic nutrition. So in that, we have already covered the saprotrophic mode of the nutrition, parasitic mode of the nutrition. So now today we'll be just doing about this Holozoic nutrition. So starting the Holozoic nutrition, if we say this Holozoic nutrition is again a mode of heterotrophic nutrition. This is again a mode of heterotrophic nutrition. Heterotrophic nutrition, which involves the intake of solid pieces of food, which involves the intake of solid pieces of food. So here again, if you see there in the saprotrophic mode of nutrition, we can see that the food is absorbed in the simpler form and it's usually in absorbable form. And in case of the parasitic nutrition, there you see the food is taken or absorbed in either it is pre-digested or it is semi-digested. But here in case of the Holozoic nutrition, things are different. Here the food is taken in solid pieces. So since this taking in food, since food is taken in, it's also called as ingestive nutrition. So it's also termed as ingestive nutrition. Since it involves the intake of food or the food is taken in, so we also termed it as an ingestive nutrition. So firstly, on the source, now the source is food. If the food is in the form of plant, or it can be in the source of an animal, or if it can be both, or it can be dead animals. So if the organisms obtain the food in plant form, or the food is from the plant or the plant itself, then we term that organism as an herbivore. They are the herbivores. If the organism takes in the food in the animal form, then we termed it as an carnivore. And if they take both, that is in plant as well as in animal form, then we say it's an omnivore. Now again, if the food is taken in the form of the dead animals, then we say that it's an detrivore. Now there is a common confusion in students that they don't understand the difference between the detrivores and the saprotrophs. So we are going first to remove that confusion, that what is the difference between a detrivore and the saprotroph. So if we try to enumerate the difference between the mode of the saprotrophic and the detrivorous nutrition, we'll see the differences. When we talk about the saprotrophs and detrivores. Here in case of saprotrophs, you can see the food is taken inside the body in an absorbable form, or we can simply say that the food is taken in simple, absorbable form. The food is digested outside the body. The food is digested outside the body, outside the body, or we can say the digestion is outside the body. So when the food is digested outside the body, the digestion is termed as external digestion. It's the external digestion. Now here in case of the detrivores, what happens? Food is taken in solid complex form, now this food is converted into simpler absorbable form, or the food is digested inside the body. The food is digested inside the body, or in simpler words we can say that the digestion is internal, or it has the internal digestion. So this was the basic difference between the saprotrophic nutrition and the detrivorous nutrition. So now our next thing will be how do Holozoic organisms obtain their nutrition. So firstly, if we see that this mode of Holozoic nutrition is usually shown by animals, and it's also shown by protozoan, or it's also shown by unicellular protists. Unicellular protists. So now the question arises, how do these animals or unicellular protists obtain their nutrition? When we talk about these unicellular protists, in case of unicellular protist, whole body surface is capable of ingesting the food. Example is amoeba. Or in certain unicellular organisms, like in case of paramecium, there are definite points for ingestion as well as for egestion. Then moving on to the animals. Now in case of animals, they have different specializations. In the form of digestive system, which comprises of digestive organs and digestive glands, and glands. So this is the mode, how do these Holozoic organisms obtain their nutrition. Now we will be substantiating this Holozoic mode of nutrition in both unicellular organisms as well as in multicellular organisms. So first of all, we will be substantiating it with the help of an amoeba. So how does amoeba obtain its nutrition? Nutrition in amoeba. So this, when we talk about nutrition in amoeba, firstly when we say this amoeba is a microscopic structure and you can see this amoeba only with the help of an aid. So amoeba usually is unicellular and can be seen with the help of an aid. So this aid is usually a microscope. So when we talk about the body organization, of amoeba, we say that it is basically a eukaryotic. It is basically a eukaryotic organism. So that means this will have, if we see the diagrammatic representation of amoeba, we say that this amoeba is a eukaryotic organism. So this basically has a well-developed nucleus. So nucleus is there and it has essential cellular organelles. These cellular organelles, it is having mitochondria is also there. Then it will be having ER, that's the endoplasmic reticulum. Endoplasmic reticulum is also there. ER is also there. And it will be having lysosomes are also there. And contractile vacuole is also there. So all the essential organelles that a eukaryotic organism should possess, that are possessed by this amoeba. Then talking about the habitat of this amoeba, when we talk about the habitat of this amoeba usually is found in the aquatic habitat. It's the aquatic or it's in moist places, aquatic or in moist places. Okay. So this was something about the amoeba. So when we talk about the Holozoic nutrition, whether it is in case of, so when we talk about this Holozoic nutrition is usually achieved in 5 steps, either in case of unicellular organism or in case of multicellular organism. The process of nutrition in the Holozoic organism is achieved in 5 steps. The first one is the ingestion, and the second is the digestion, and third is the absorption, and fifth is the assimilation, this is the first, this is the second, this is the third, and this is fourth, and fifth is the egestion. So when we talk about the Holozoic nutrition, this is basically achieved in 5 steps in case of unicellular organisms and in case of multicellular organism. So the 5 steps are ingestion, digestion, absorption, assimilation, and egestion. So let's talk about first this process of ingestion. So if we see the amoeba, this is the amoeba. This amoeba has a speciality that its membrane outside is it has the external flexible cell membrane. So when we talk about the cell membrane of the amoeba, that is flexible, and with the flexibility of the cell membrane, it is able to form special finger-like projections. It is able to form finger-like projections known as pseudopodia. So as I said that this flexible membrane produces finger-like projection, and these finger-like projections, like I'll be showing it, if we see this membrane is flexible, it can produce the finger-like projections like this. These finger-like projections are produced because of the flexibility of the membrane, and this flexibility of the membrane results in the finger-like projection known as a pseudopodia, and these pseudopodia helps in the movement. So these pseudopodia helps in the movement. Now another question is, here it's an eukaryotic organism, so it lacks eyes, everything. So the question is, if the food is in the surrounding medium, suppose this is the food, how does this amoeba comes to know that food is there? Because if you see in multicellular organisms, they have certain specializations in the form of the eyes which can locate the food. Here in case of amoeba, it does not have eyes or anything by which it can locate. So what makes it to move towards the food or to proceed towards the food? So if we see, firstly, when we talk about this food, as amoeba itself is a unicellular organism, what does its food comprise of? So when we talk about the food, we say this food is usually another unicellular organism. Food is another unicellular organism. Now this unicellular organism might be in the form of the animal, or it can be in the form of a plant, or their parts. So talking about the food, what does the food comprise of? The amoeba when it takes the food, what is the food that comprises of? When we talk about the food, we say that the food is in the form of the animal, or it's in the form of the plant, or it's their parts. Now when the food is there in the surrounding medium, as we know, there are certain chemicals that are released by this food. If this is the food stuff, there are these certain chemicals which are released by this food. And when we talk about this amoeba, we say that on the cell membrane of the amoeba, there are small receptors. There is the presence of small receptors. So when these chemicals that are produced by this food, they diffuse in water, say these are the chemicals. These are the chemicals that are released by this food stuff that are diffused. Now these chemicals, there in the water or in the moist conditions, it is diffused, and once this is diffused, and it reaches towards the receptors. So firstly, what happens? The chemicals are released. The chemicals are released by the food. And then these chemicals diffuse in the medium. Now once these chemicals diffuse in the medium, they bind to the receptors that are present on the membrane of the amoeba. Then these chemical binds to the receptors. these chemicals are being released and they here bind to the receptor, they bind to the receptors. Now, when they bind to the receptors, we say these receptors are then stimulated. The receptors are then stimulated. So, these chemicals, they act as a stimulus for these receptors. Now, once these receptors are stimulated, then once these receptors are stimulated, once receptors are stimulated, amoeba moves towards the food. Amoeba moves towards the food, moves towards the food with the help of pseudopodia. With the help of pseudopodia. With the help of pseudopodia. Once again, I'll tell you about when we talk about the stimulus. When we talk about the stimulus. So here I say that these receptors needs to get stimulated before they move towards the food. So the stimulus. So talking about the stimulus can be in the form of the chemical or it can be in the form of the light. So these receptors, once they get activated with the chemicals, they will move towards the food, and that movement towards that food is termed as chemotaxis. So the movement which has been brought down with the help of the chemicals is termed as chemotaxis. Now the second will be light. If the movement of the amoeba towards the food source is with the help of the light or is produced with the help of the light, then it's termed as phototaxis. So now this concept is clear that the movement of the amoeba towards the food is produced either by chemicals that are released by the food itself or either by the light. So if it is chemical, then the movement is brought down by the chemicals, it's termed as chemotaxis, or if the movement is brought down with the help of the different light intensities, then it is termed as phototaxis. Now let's see what happens after that. Now once this amoeba reaches towards the food, now this is the food stuff. Now this amoeba reaches towards the food with the help of the stimulus. Firstly, what it does, it encircles the food. Firstly, it traps the food, then it encircles the food. This is the food or this is the prey, it encircles it. Then, once it reaches towards the food, it encircles it. It encircles it with the help of plasma membrane. Once this food is encircled, it results in the formation of a vesicle. The food is trapped inside this vesicle. Now the food is trapped inside the vesicle and this vesicle gets pinched off. Now what happens? It encircles the food and with the help of the plasma membrane. Now what happens? What is the second step? The vesicle formation takes place. The vesicle formation takes place and the vesicle gets pinched off. And the vesicle gets pinched off, and the plasma membrane fuses again. Now we can see here, now this vesicle gets pinched off. Once it gets pinched off, so this has gone pinched off, now it is inside the amoeba or it is inside the cytoplasm. Now here we should remember that the process by which the food moves across the cell membrane or goes inside the cell membrane, that process is termed as endocytosis. That is termed as endocytosis. Once again, what is endocytosis? Endocytosis is the process by which the food is taken inside the cell membrane by the formation of the vesicle. Now this endocytosis can be further classified if the food taken is in solid form. If the food taken in is in solid form, then the process is termed as phagocytosis. Then the process is known as phagocytosis. In simpler terms, this phagocytosis means cell eating. This phagocytosis means cell eating. Now, if the foodstuff is taken in the form, if it is in liquid form, then the term we use is pinocytosis. So pino is some in liquid form and cytosis is taking in. So we say that it's cell drinking. It's cell drinking. So now the process by which the food is taken inside is termed as endocytosis, and endocytosis usually can be of two types. If the food is taken in solid form, then we say it is termed as phagocytosis. Now if the foodstuff is in the liquid form, then the process is known as pinocytosis. Now again, when we talk about this endocytosis comes under the bulk transport. This is basically the bulk transport. And this bulk transport is energy dependent. Now the vesicle formation takes place and the vesicle gets pinched off and it moves there inside the cytoplasm. So this part is basically the ingestion. So now what is ingestion? If we say in ingestion comes that it's moving towards that particle, food particle, it encircles it, it fuses, the plasma membrane fuses and the vesicle gets pinched off and it moves towards the cytoplasm. Then the next step is basically the digestion. Now in the second step, digestion, what happens? This vesicle that is formed, this vesicle is also termed as carrier vesicle. This is also termed as carrier vesicle, or this is also termed as phagosome. So you can use different technical terms for it. Either you can call it as a vesicle, either we say it's a carrier vesicle, because why we call it as a carrier vesicle? Because it carries, see, inside its food and outside is the cowring. Now this cowring is same as that of plasma membrane. The cowring or the composition of this cowring that surrounds the food is same as that of plasma membrane. So we say it also consists of double membrane. This is double membrane envelope. So if a question comes like, what is phagosome? Phagosome is basically a carrier vesicle that carries the food or the prey, and that is encircled by the double membrane covering, and this covering, the composition of this covering is same as the plasma membrane. Now once it's gone in, so inside it you will find it's inside of the amoeba is filled with the liquid substance, and this liquid substance is known as protoplasm. Now this protoplasm, if we see this protoplasm is in continuous perpetual motion. So when we say this protoplasm, this is in continuous perpetual motion. So we say this cytoplasm inside is not stationary, it is not standstill, it is in continuous perpetual motion. That means it flows slowly. So this continuous perpetual motion, we also give a technical term to it, we say it is termed as cyclosis. So once this gets pinched off and it moves towards the protoplasm, with the help of the streaming movement, with the help of the cyclosis, this carrier vesicle is taken in. This carrier vesicle is taken in. Now this is the carrier vesicle. This carrier vesicle is taken inside the cytoplasm. So firstly what happens? Takes in the carrier vesicle, and now what is the second step? The second step is there, as I told you, the organization is eukaryotic. Then inside what happens? A lysosome gets attached to this phagosome. What is the second step? Second step is lysosome gets attached to the phagosome. First step was the cytoplasmic movement or cyclosis. That cyclosis brought the phagosome in, and then a lysosome got attached with this phagosome. Now firstly we should know that what does this lysosome contain. When we talk about this lysosome, we say this lysosome basically contains 50 different types of enzymes, or it contains 50 different enzymes. So this lysosome got fused with the phagosome and it resulted in the formation of temporary stomach, or food vacuole, or if we give a technical term to this temporary stomach, we call it as an gastrule. So if a question like this comes in the board examination, that what is basically a gastrule or what is the structure of the gastrule, in simple terms, gastrule is basically when the phagosome fuses with the lysosome, it results in a structure that is termed as the gastrule. Then what is the next step? When this lysosome got fused with this phagosome, this lysosome releases enzymes. This lysosome releases enzymes inside the phagosome. So the next step will be Next step will be lysosome. Lysosome will release enzymes inside there in phagosome. Now, once these enzymes are released there in the phagosome, what happens? The complex food with the help of these enzymes, will get converted into simpler form. This will get converted into simpler form. So, this will get converted into simpler form. Now, once the food is converted into simpler form, then what happens? The digested simpler food gets diffused out. So, once the food is converted into simpler form, then the simple food will diffuse out of the carrier vesicle or the gastrule. Then next step will be simple food. Simple food will diffuse outside When this food will diffuse outside. So, this was the whole process. The process of lysosome fusing with the phagosome and resulting in the formation of the gastrule, and then the lysosome releasing the enzymes, and then afterwards the conversion of the complex food into the simpler food with the help of the enzymes released by the lysosome comes all under the digestion process. So, digestion was basically, if we try to sum up this digestion, so this digestion you can sum up, first step will be the fusing of or the fusion between the phagosome and the lysosome, and the second step will be that the lysosome is releasing the enzymes and there the complex food is being converted into the simpler food with the aid of or with the help of these enzymes, and the third step will come there in the that the simple food is being diffused out there in the cytoplasm again. So, now there will be the next step. So, the next step will be the absorption. So, now what happens there in the absorption? Firstly, this diffused food which came out of the gastrule diffuses throughout the cytoplasm with the help of the cyclosis. So, what happens here? Simple food gets distributed throughout the cytoplasm via cyclosis. So, now the food that is released, it is being distributed. It goes here, everywhere. It is distributed throughout the cytoplasm. Then what happens? Next step will be this simpler food will be absorbed by the various cellular organelles for their biochemical reactions. So, this simple food will be absorbed or taken in by various cellular organelles, for performing their various metabolic activities. So, this all comes under absorption. In absorption, firstly, the simple food is diffused out there in the cytoplasm, then it spreads throughout the cytoplasm with the help of the cyclosis. Then this simple food is absorbed by various cellular organelles for performing their biochemical activities. Then this absorption is followed by another process that is termed as assimilation. That is termed as assimilation. Assimilation. Now, what happens there in assimilation? In assimilation part, the food that is taken up, that is taken up by various cellular organelles, the food that is taken up by the various cellular organelles is utilized for biochemical activities. So, with the help of these biochemical activities, the protoplasmic mass is built up. The protoplasmic mass is built up. Either it is via the catabolic activities or the anabolic activities. In simpler terms, we say that assimilation is basically the building up of the constituents of the protoplasm. When various constituents of the protoplasm are synthesized with the help of the cellular organelles as they consume the simple form of food, that is termed as assimilation. Now, once the food has been assimilated, what happens to this vesicle? Now, the last step is the egestion. Now, talking about the egestion, what happens there in the process of egestion? Lysosomes are detached. Lysosomes are detached from phagosome. Lysosomes are detached from the phagosome, or either they are disintegrated, or lysosomes are disintegrated. And now this food vacuole contains only undigested food. Then this food vacuole contains only undigested food. Now this food vacuole, when it contains only undigested food, it gets heavier. So, when this food vacuole contains only undigested food, it gets heavier. And via cyclosis, it moves up. And via cyclosis, it moves towards the cell membrane. Now, see children, this part is very important, as you know that the process by which the undigested food is thrown out of the cell, we usually term it as exocytosis. But what happens there in the exocytosis? This is somewhat in children it seems to be unclear. So firstly, let's try to see what happens when this undigested food vacuole reaches near the cell membrane. Now suppose this is the amoeba and this is the food vacuole which contains undigested food. It first reaches towards the cell membrane. So firstly, when it reaches towards the cell membrane, during the process of exocytosis, it fuses with the cell membrane. So first is that it fuses with the cell membrane. It fuses with the, sorry, not cell membrane, plasma membrane. It fuses with the plasma membrane. It fuses with the plasma membrane. Once it fuses with the plasma membrane, it either gets incorporated, after fusing it gets incorporated, it gets incorporated in the plasma membrane. Once it gets incorporated in the plasma membrane, the undigested food is thrown out. So the undigested food is thrown out with the help of the process that is known as exocytosis. And then this, now there is always a curiosity in the minds of the children that what happens to this food vacuole. This food vacuole which contained the undigested food becomes the part of this plasma membrane. It becomes the part of this plasma membrane, it gets merged with it, it gets incorporated there in the plasma membrane and it becomes the part of the plasma membrane and the food is thrown out via the plasma membrane. So this whole comes under the process of the digestion. So once again, if we try to summarize their process of Holozoic nutrition in the amoeba, we might sum up it in 5 steps. So the first step is basically the digestion, where the amoeba locates its food and it moves towards its food with the help of the chemotaxis. With the formation of the pseudopodia, it just encircles the food and takes it inside the plasma membrane with the help of the process that's termed as endocytosis. Now, if the food is in solid form, then the process of endocytosis or the subtype of the endocytosis, we termed it as an phagocytosis. Or if the food taken inside the membrane is in liquid form, then we say it is pinocytosis. So basically the first step of the Holozoic nutrition is ingestion. So ingestion is simply the taking in of the food substances via the process of endocytosis. Now the second step is the digestion. Now what happens there in the digestion, that as the food is taken inside the plasma membrane, it is encircled by the part of the plasma membrane itself and it results in the formation of a structure, and that structure is termed as phagosome. Now this phagosome, once it enters the cell membrane and it is at that moment it is towards the periphery only, then with the help of the cyclosis. Now what is cyclosis? Cyclosis is basically the perpetual motion of the cytoplasm or the protoplasm inside. Then with the help of the cyclosis, this phagosome or the food particle which is inside the carrier vesicle is taken inside. And what happens then, the second process, that is the digestion, takes place. What happens there in the digestion? This phagosome basically gets attached to the lysosome and it results in the formation of a temporary structure, and that temporary structure is termed as gastrule or temporary stomach. Then what happens there in the temporary stomach? As this lysosome contains 50 digestive enzymes or 50 enzymes, it sends off or it secretes its enzymes there in the phagosome, and inside the phagosome what happens? A chemical reaction takes place. The food that is in the complex form, it gets converted into the simpler form. Once the complex food gets converted into simpler form, it gets diffused out of the membrane. Once it gets diffused out of the membrane or outside this temporary stomach, what happens then? It reaches there in the cytoplasm with the help of the cyclosis. Now remember, once the food diffuses out of the temporary stomach, here the digestion ends. Then the third step, that is basically the absorption starts. What happens there in the absorption? This simpler food basically travels with the help of the cyclosis and it reaches towards the points where the food is required. Now, the points may be in the form of the cellular organelles. Now, these cellular organelles, they take up this simple form of food and they take in and then they start the biochemical reaction. Now, basically the whole part of the biochemical reaction where this simple food has been absorbed by the organelle and it comes under there in assimilation. Now, what happens in assimilation? The absorbed food that has been absorbed by the cellular organelle gets converted into different stuff or into different structures. Basically, what are those structures? Those structures basically increase the protoplasmic content of that cell. So, as you know, amoeba is cell. So, basically what happens there? This assimilated food gets converted into different protoplasmic structures and it results in the increase in the protoplasmic content. So, this process or this part comes in the assimilation. Now, what happens there in the digestion part? In digestion part, the leftover food or the undigested food that is present there in the food vacuole gets heavier and it rises up to the plasma membrane. Now, once it rises up to the plasma membrane, what happens there? There many confusions are always there. What happens? Firstly, it gets incorporated with the plasma membrane. Then once it gets incorporated with the plasma membrane, it becomes the structure of the plasma membrane and it throws the food particles, undigested food particles out and it merges with the plasma membrane only. So, this process is known as digestion. So, this was all for today. That today we have seen the Holozoic nutrition there in the amoeba. In my next lecture, we will be seeing the Holozoic nutrition there in the humans. Thank you.